Ma'am, you are on live.
Good morning to one and all. On behalf of Department of Biotechnology, Dr. NGR Educational and Research Institute, we welcoming you all for 6th-day International Alumni Series Roadmap to Hybrid Research 2. Now, I'm very much excited, feeling happy to welcome Mr. Aakash Nagaraj, our proud alumni. He has completed an undergraduate in biotechnology in Dr. NGR Educational and Research Institute. Currently, he is pursuing his master's in University of Alabama, Birmingham, USA. Aakash, thanks for accepting our invitation. Now, Aakash, the stream is yours.
Yeah, thank you, ma'am. First of all, I would like to thank the Department of Biotechnology for this opportunity to give this talk, and all the faculty organizing this, HOD, the deputy HOD, and everyone. So I will start. Sir, can you see my screen? My PowerPoint screen?
No, Akash, we couldn't see it.
No, pa. You just give the share screen option.
Share screen in the beneath. Yeah,
Okay. Now I can see.
you can make it in a presentation mode.
Okay. So my topic today is genomics and its application in health and disease. This is just a short overview of what I'll be talking today. First of all, introduction. What is DNA? Just basics and the difference between genetics and genomics. Then the subfields of genomics, little bit of history, and about the Human Genome Project, why to sequence genome, uh, then DNA sequencing and its reducing cost, and then the different omics technologies, and finally all the applications of genomics in different uses. Like I'll be talking about 4 uses: disease diagnosis, crop development, prenatal testing, and vaccine and drug development. So first, a bit of introduction about genomics. It is the study of whole genomes of organisms. It is focused on the sequence of the genome and the structure, function, evolution, mapping of the genome. In the left-hand side, we can see an image of DNA. It's the double-stranded DNA, and in the bottom image shows a karyotype of the human genome. So human beings have 23 pairs of chromosomes, and 22 of those are autosomes, and one is sex chromosome, one pair of sex chromosomes. So all of our DNA is the genetic material is stored in these 23 pairs of chromosomes, and we get one chromosome from each parent. Suppose chromosome 1 means there is a pair, and one is the paternal chromosome and the other one is the maternal chromosome, and we get one from each parent. So what is genetics? It's the area of that's concerned with primarily sequence organisms genome. This could be any organism, humans, plants, animals, and also microbes. It involves the study of genes at the DNA level and at the messenger RNA and at the proteome level of both single cells and the whole tissue. This term was first coined in 1986 by a scientist Tom Roderick at the Jackson Laboratory during a meeting while they were discussing about mapping the human genome. This is a bit of brief overview about DNA. Some people would have already learned this, and some people won't know. So, DNA is a double-stranded molecule, as we can see in the left-hand side of this image. It is made up of three components. It has a sugar component, a phosphate group, and nitrogenous base. Here in the bottom image, you can see the three parts of a nucleotide. So DNA is made up of nucleotides, and here we can see the phosphate group. And next is the pentose sugar, which is a 5-carbon sugar, and that pentose sugar for DNA is deoxyribose sugar, and that is how the DNA gets its name, deoxyribonucleic acid. And the third component is nitrogenous base. There also could be 4 variants of nitrogenous base in each nucleotide, either adenine, guanine, or cytosine, or thymine. And so the 2 strands are made up of nucleotides, and they are connected at the nitrogenous base. In the top image, we can see they are bonded at the nitrogenous base with the help of hydrogen bonds. And after bonding, they will intertwine against each other to form a coil like this. So we know that DNA has all the genetic information of the cell, and the cell must get the information to use it. And how does the cell get the information? So it uses 2 processes to get the information: transcription and translation. These two steps are the goal is to produce proteins. So proteins are the main workforce of the cell. So all of the functions of the cell is done by proteins. So this DNA goes to messenger RNA, mRNA, and that goes to protein. So this is known as the central dogma of biology, and this is true for all cells of all organisms, from bacteria to even large animals. So how all cells get the information is through DNA, and that gets converted into messenger RNA and then to protein. And we also see an image here, the double-stranded DNA, and that getting converted into a single-stranded RNA molecule. And using the RNA molecule, a polypeptide chain is produced, protein. This slide also describes about the transcription and translation process. So the mRNA produced at the nucleus, that is transported from the genome to the ribosome. So the messenger RNA is produced at the nucleus. That's where all the DNA is. And this image shows the description of the process. So the 2 strands are uncoiled by enzyme, and then enzyme known as RNA polymerase reads through the template strand, and that produces a messenger RNA strand. And then the messenger RNA strand, which is produced in the nucleus, migrates to the ribosome. So ribosome is the protein factory of the cell. That is where all the proteins of the cell are produced, and that is where protein synthesis also takes place. So at the ribosome, here's a picture of the ribosome, and here we can see an messenger RNA strand which goes through the ribosome, and based on the sequence of messenger RNAs, a complementary polypeptide chain is assembled, which is made up of amino acids. So that is how proteins are produced, and once the entire messenger RNA is read, one polypeptide chain or a protein is produced. Let's see a little bit about the difference between genetics and genomics. So genetics is specifically the study of single genes. So genetics is used to study the function of a particular gene. We sequence the gene and study the gene using a variety of tools, while genomics on the other hand refers to the study of all of the organisms genes together. It is more kind of a comprehensive study. So genetic studies the function of a single gene and the composition of the single gene and all its properties, and genomics addresses all the genes together and their interrelationships. This slide shows the different subfields in genomics. There are 4 subfields: functional genomics, structural genomics, comparative genomics, and epigenomics. So functional genomics is the characterization of the genes and their messenger RNA and the protein products which is produced. So this is basically finding out the sequence of the gene and which protein is produced, so on. Structural genomics is concerned with the dissection of the architectural features of the genes and the chromosomes, like the structure and how it is positioned, stuff like that. And comparative genomics is the study of the evolutionary relationships between genes and proteins of different species. So we can compare genes of different species such as human beings and other primates, suppose chimpanzee or other primates, or even different plants or different bacteria, to see how the different genes have diverged. And then the 4th subfield, epigenomics, that describes, that studies the DNA methylation patterns, imprinting, and DNA packaging. So the DNA methylation controls the activation and inactivation of many genes, and DNA packaging is how the entire of genetic material is compressed into the nucleus, that is with the help of proteins known as histones, and that is also one subfield. Let's see a little bit of history, just short about all the techniques which were developed. So Fred Sanger was the first scientist who to sequence the complete genome of a virus and then a mitochondrion. He did that in the early 1970s. And then one more scientist known as Walter Fries in 1972, his group was the first to sequence a single gene. They sequenced the gene of bacteriophage. And then in 1995, one more scientist known as Hamilton O. Smith and his team were the first to sequence a genome of a free living organism known as Haemophilus influenza. So by 1995, the first genome was sequenced of an organism, and during that time, Human Genome Project was already running. I will talk about that in the next slides. So now let's look at our genome, the human genome, the size of it. So human DNA or the genome consists of a total of 3 billion base pairs, and more than 99% of the base pairs are similar in all the people. There is only a slight variation in certain sections of the genome sequence, and SNPS or single nucleotide polymorphism are a common type of genetic variation found in different people. So our human genome was first, the complete genome was sequenced in, the first draft was published in year 2000, sorry, excuse me. And around 20,000 protein coding genes were discovered at that time when we got the sequence of the human genome. And the size of one genome file would be around 80 to 90 GB. That's because that's very large. Now let's look at why we need to sequence genomes, what is the purpose of it, and what we can learn using the genome sequence. So sequencing genomes helps us to understand how the genome as a whole and how the genes work together to direct the growth, development, and maintenance of an entire organism. And the genome sequence could also be used as a shortcut to find out all the many more genes than we know currently easily and quickly. So after we sequence the genome, we were able to find out our human genome of 20,000 genes. Now let's look at the Human Genome Project. So this project was an international and collaborative research program of many different countries, and the research institutions, the government research institutions of particular countries, and many universities too. And the goal of this was the complete mapping and the understanding of all the genes of human beings. So this project was officially launched in 1990, and the final draft of the human genome was published in 2003. The cost of this project, the final cost was cost $3 billion. It was very expensive to finally just sequence a single genome at that time 20 years ago. And now let us look at the cost, how the cost is reduced now after 20 years, after the development of many better, easier technologies. The cost has reduced dramatically, and the current cost of sequencing a genome is less than $1,000, and that cost is reducing and will reduce in the future. This is a little bit about DNA sequencing. So DNA sequencing means finding out the exact order of bases in a strand of DNA. One of the first method of DNA sequencing, one of the first good methods was Sanger sequencing, that was developed in around 1975, And then a better variant was produced known as Maxim Gilbert sequencing in around 1977-1978. And the technique used for the Human Genome Project, that was the shotgun sequencing method. And then in recent years, in the past decade, high throughput methods have been developed, known as next generation sequencing. These methods, next generation sequencing, has dramatically reduced the time to sequence the genome and also the cost. So let's see a little bit about shotgun sequencing. So this was used in the Human Genome Project to sequence a genome. So there are two phases for this, and the first phase is known as the shotgun phase. In this, the genome is divided into numerous smaller DNA segments of appropriate size, and after subdividing into smaller overlapping, and after subdividing it, and these smaller fragments were then sequenced. These smaller fragments were also overlapping. And then the second phase of this project was known as the finishing phase. So after the many of these smaller fragments were sequenced, all these have to be assembled into a single genome in a linear order. And there are also many gaps, and so the second phase was in assembling it and filling up these gaps and resolving the DNA sequences in ambiguous areas. Here is a graph of how the cost has reduced to sequence per genome. It started at $100 million, and it's been reducing dramatically. And this chart stops at 5 years ago, and the past 5 years also it has kept on reducing. In this right hand side image shows the couple of famous sequencing machines manufactured by certain companies. Alumni, that's one of the commonly used, and it's one of the best. All these are next generation sequencing machines. And also you can see the runtime here. Previously it used to be more than 24 hours and maybe even days, but now this with newer machines the time is also being reduced 2 hours and 45 minutes. Now I'll talk about omics technology. So this is derived from the word genomics, so GE, and then omics. So this is based on sequencing, and we can sequence not just the DNA and the cell, but certain other things too, which play an important role. So we can sequence also messenger RNA, and then the proteins which are produced by the cell and the metabolites. So sequencing mRNA would be known as transcriptomics, and proteins is known as proteomics, and metabolites is known as metabolomics. These are the four main omics technologies, and they have very broad range of applications. So mass spectrometry is a method, and that is the most common method used in proteomics and metabolomics. That is the machine which is used to get the data. And for messenger RNA, it's known as RNA-seq. And nowadays, recently we can also sequence a single cell RNA-seq has also come into play. So sequencing genes, it's genomics, mRNA, transcriptomics, proteins, proteomics, and metabolites, metabolomics. So we can get all these 4 types of data from a specific biological sample. Suppose we can yeah, take blood from an individual, and we can get all these 4 types of data, or in other animals or tissues or bacteria or any kind of biological sample. This slide also basically mentions the same thing as the previous slide, but this shows, like, first is the gene, and then gene information from the gene is converted into messenger RNA. We can also sequence that to see which of the messenger RNA is produced by a particular tissue, say liver or heart or lung. If we sequence the messenger RNA, we can find out which are the genes which are being expressed in that tissue. And then we can do proteomics. So from the messenger RNA, that would be converted into protein. So if we do proteomics, we can know the information about the proteins which are produced by the cell or the tissue. And similarly, metabolomics, all the different metabolites which arise due to the cellular chemical reactions taking place. These slides get more in detail about it. So first is genomics. That's the most mature of the omics field. It was the oldest. So this is focused on identifying genetic variants associated with disease and response to treatment or future patient prognosis. So sometimes certain diseases may be associated with certain genetic variants. One common example is cancer. So cancer arises due to gene mutations, and there will be a patterns for certain types of cancer. Certain types of cancer arise only due to a particular mutation, and using genomics we can get all the information about that cancer. And also response to treatment. Different patients will respond differently to treatments, and genome and their genetic sequence might play a role in that. Next is epigenomics. So this focuses on the genome-wide characterization of the modification in DNA, that is the reversible modifications which take place. DNA methylation. Methylation is the addition of methyl groups to the DNA, or histone deacetylation, histone acetylation. So this is addition of certain chemical groups on the surface of the DNA, and once these methyl groups or other groups are added on the DNA, this changes the gene expression of the genome. So certain genes can be expressed, highly expressed, and certain genes can be will be turned off. It's like an on and off switch. This epigenetics is a new field. Basically, we are still learning about it, and it's come into prominence in the past, maybe primarily in the past 10 years. And this has been associated with many diseases, variety of diseases, all the major diseases. There is some changes in epigenetic change in the cell tissue. So next we look at transcriptomics. This is sequencing the messenger RNA in a cell. This is also one of the widely used tools in research, knowing the gene expression of the cell or tissue type. So using this, we can know all the genome-wide RNA levels, whichever RNAs are produced by a particular cell. We know that there are 20,000 genes, and out of that, only a certain only a small subset of these genes will be converted into messenger RNA. So we can study that both qualitatively and quantitatively. Qualitative meaning we can find out which are the transcripts which are present in that particular sample, biological sample. And we can also identify novel splice sites and RNA editing sites. And quantitative meaning we can find out how much of each transcript is present. Some in some tissues a particular gene may be highly expressed, and that means that particular mRNA transcript would be in very much higher levels, suppose 100 times more or 1,000 times more. Next is proteomics. This is sequencing all the proteins in the cell. This is used to quantify the peptide abundance, its modification and interaction. Next is metabolomics. This simultaneously quantifies all the multiple small molecules, the amino acids, fatty acid, carbohydrates, and other products of cellular metabolic functions. Next is microbiomics. This is studying the microbes living in our body. There are many different microbes live in our body, especially it is concentrated in the human gut, which is our large intestine. So in our large intestine, not just humans, in all animals, a lot of bacteria live. The amount of the total number of bacterial cells is almost more than the human cells. That's the amount which is present. It is a lot. We still don't know accurately how much microbes there are, and using this we can sequence the genomes of all these microbes, and sequencing that we can know the information about all the different types of bacteria present, not just bacteria, also viruses and fungi. So for the first part, we saw all the different techniques of genomics, and now we can see its application. It has numerous applications. Here are some examples I have put in the slide. First is molecular biology. We can study genomes and the proteins they encode. Then it's evolutionary biology, studying how different organisms evolved and their relationship. Then it's metagenomics, finding out the different organisms present in different, suppose water, sewage, dirt, and air, or we can swab any other samples from other organisms. Then in medicine, like we can sequence genes and genomes and of the patients and find out their risk of genetic diseases. And one more use is in forensics. I will talk about 4 of the uses in slightly more detail. So first is prenatal testing. So this is to find out problems in pregnancy very early on, or even before the conception of the fetus. So there are 2 methods. There are both invasive and non-invasive method in this. Some non-invasive is using cell-free fetal DNA. This can be, we can get this from the blood of the mother, and by sequencing the DNA of the fetus using shotgun sequencing or other techniques, we can determine many things, whether if there is a problem such as mutation in the chromosomes and single gene disorders. This could be used to find out problems and maybe prevent it early on. This prenatal testing could also be used on the suppose father and mother to see whether if they have if they will have any problems conceiving a baby, and whether there are certain genetic disorders which a baby can have, such as Down syndrome, sickle cell anemia, thalassemia, cystic fibrosis, and muscular dystrophy. So using prenatal testing, we can find out the risk of these diseases, and we can take measures, whatever kind of measures they are, to prevent trouble later in life, which might cause the death of the newborn child, or which might cause trouble later on in the future. And one more use is in disease diagnostics. So by sequencing using genomics, whether sequencing a genome, we can find out our risk of getting certain diseases. One of the examples which I have shown here is cancer. So there's a particular gene known as BRCA gene. So BRCA1 and BRCA2, these two are 2 of the tumor suppressor genes. And women who have mutations in this, either of the BRCA1 or BRCA2 genes, have a higher risk of getting breast cancer and other type of cancers too. For breast cancer, it is 5 times the risk, and ovarian cancer, it is 10 to 30 times. Here is a image of where the genes are located in the chromosome. One of the genes is in chromosome 17, and the other is in chromosome 13. This image shows a simple representation of how the mutation could be passed on from the parents to the children. Having the mutation doesn't mean that the person will get cancer. It just means that they have a higher risk of getting cancer, and they can take early action, like preventive measures, like they can do screening, yearly screening, and other things. And they can also go for surgery, mastectomy, and other ovary fallopian tube surgery in case they have such a high risk of cancer and they don't want to risk it. And the next application which I'm going to be talking about is in crop improvement. So this is also a very big field in agricultural genomics, to get this sequence of our crops which are used in agriculture, many different crops. This could be used to improve the product productivity and the sustainability in crop and livestock production. There are 4 points given here: comparative genomics for plant breeding, transcriptome analysis for plant breeding, genotyping and marker assisted breeding, and recombinant DNA technology. So after sequencing and getting the information, we can increase production, lower the cost, and prevent crop damage due to pests and diseases. There could be many ways we can do it. One way is through finding out specific genes which might cause a disadvantage and removing it, or adding different genes that would come under the category of genetically modified organisms or GMO crops. That is still not yet widely used, and there is also controversy, or it's a big topic, like the government agencies have to, they regulate it and it has to be approved, and it's a big topic, but it has both advantages and disadvantages doing that. Here are some of the example genes. So genes for herbicide tolerance could be put in plants, and pathogen resistance, pest resistance, Bt genes, quality improvement, fertility restoration, chilling resistance. And each of these would have a specific use, and if it is done, there will be we can know the crops won't die of pests, they will be pest resistant, and the overall production will also increase, and the quality also could be increased. So there will also be more profit for the farmers. This just shows a quick timeline of genome sequencing in plants. Not much in the slide. First was rice was sequenced in 2002, then maize, then soybean in 2010, cabbage, potato, barley, tomato, and then peach, orange, chickpea in 2013. And one more use is in vaccine development. Vaccination are basically one of the most effective tools for the prevention of infectious diseases. And the availability of the genome sequence of the pathogens, suppose it is a bacteria or a virus, and if you know the genome sequence, it will be very useful. Using such as functional genomics and structural genomics, we can find out the genetic information and also the structure of, say, proteins in the cell wall, that is where the immune system will find out to make antibodies. So by knowing this structure, by knowing the structure and genetic sequence, the genome sequence, we can have accelerated vaccine development more quicker. Usually this process is very long. The average time for a successful vaccine to come out is more than 5 years, the current average. And for the recent COVID-19 pandemic, there is urgent need, and a lot of countries are aim to develop a quick vaccine. So the next application part is drug discovery and development. So the some of the common causes of fatal diseases have been identified by genomics, and we can also identify the individuals who are uh, who have more chances of getting the particular disease before the disease even becomes apparent. So genomics has positively impacted drug research strategy and the drug discovery process. And after using the various omics tools, genomics tools, the rate of failure of drugs in clinical trials have also reduced, which have saved precious time and money. The current timeline for getting a particular drug out is, I think, almost 8 years for a successful drug from preclinical stage to the final stage, and most of the prospective drug candidates will fail in the stage. And by using genomic technologies, by getting more information from the particular drug candidates, the rate of failure can be reduced. So this image shows us the two different ways of how scientists study disease and to find out the causes of disease, and the next step would be to make drugs. So the old way was just getting a sample and studying its pathology and causes. Now we can take a particular biological sample from a patient tissue sample, and then we can do a lot of sequencing to find out the proteins, different proteins which are expressed, then get the genetic sequence, get the messenger RNA sequence and metabolomics. So by studying all this data, we can find out where there's a defect and all the changes which is caused by the disease, and we can also find out targets for the disease. Most of the drugs usually act on particular enzymes in the cells. So enzymes are proteins, and that will be useful to know to develop drugs. So this shows us the various subfields of genomics which is used in the drug development process: genome-wide sequencing and finding out single nucleotide polymorphisms, SNPS, then global messenger RNA profiling, then the protein. This is similar to what I mentioned in the previous slide: protein structure, protein expression, different proteins which are expressed, and the interaction between proteins, protein-protein interaction. And then the final stage would be industrial scale design and testing the drugs. And this is one more example of the whole process. This is also the last slide. So first we find out the disease, and then we isolate the protein, which is the malfunctioning protein which might cause the disease. So we will find out the structure, and most of the drugs target proteins. So drugs, small molecule candidates are designed by molecular modeling. First will be preclinical testing in cell cultures, and then we would move on to animal studies in mice or in other animals, and then we will scale up, then human clinical trials, clinical trial 1, trial 2, and trial 3, and finally the drug approval. And this entire, especially the first half of the process, could be significantly quicker, and rate of failure could be reduced using the various genomic tools. And that's it in my presentation. Thank you.
Thank you, Aakash. It was an amazing presentation. Thank you for giving us deep insights about genomics and its relation in human diseases and its other varied applications. We would like to convey our wishes for all your future endeavors.
Yeah,
Dear all, we have completed our 6 days of Alumni series. Heartfelt thanks to everyone to make this event more vibrant and successful. Dear participants, feedback links are posted in the description box and comment section. Thanks for traveling with us.
Ma'am, Gomadi ma'am, just unmute yourself. Hello? Ma'am, எடுத்துட்டேன். நீங்க பேசுறீங்க ஒரு வாட்டி.